Good evening. My name is Professor Moira O'Brien and I have the privilege of being the Dean of the Faculty of Science and the privilege of welcoming you to the University of Melbourne DC Blood Oration for 2023. I begin by acknowledging that we are presenting this event on the lands of the Wurundjeri people. I also acknowledge the traditional custodians of the various lands from which our online audience members are joining us today. I pay my respects to elders past and present, and I particularly welcome any First Nations people joining us here today. Thank you very much. I also acknowledge the enduring relationship with and the profound understanding of animals evident throughout First Nations knowledge systems. I also take this opportunity to call out the many members of the Melbourne School of Veterinary Sciences in the room and online. They are a magnificent group of people who have had a rough few years, so it's a pleasure to actually see you here in person. And while I cannot undo history, I can express my gratitude and admiration for their dedication to the doctorate of veterinary medicine, the discipline of veterinary sciences, and the way they have integrated into the Faculty of Science. In the 7 months the vet school has been part of the Faculty of Science, they have earned my utmost respect for what they do and who they are. It is my pleasure to have them as part of the family of the Faculty of Science, so thank you. Presented each year by a renowned veterinary scientist, the DC Blood Oration honours the legacy of Professor Douglas Charles Blood. As Dean of the then Faculty of Veterinary Sciences from 1962 to 1968, Professor Blood founded this university's modern veterinary school. I will now hand over to the current guardian of Professor Blood's legacy, Head of the Melbourne Veterinary School, Professor Josh Slater, to tell you more about this evening's event and our distinguished guest speaker. Thank you, Josh.
Moira, thank you. Distinguished guests, blood family members, Dean O'Brien, alumni, donors, supporters, friends, colleagues, DVM students and research students, welcome to the 2023 DC Blood Oration. It's my honour and privilege to welcome you to this important event in our school's calendar. And thank you very much for joining us in person at the Woodward Centre. And again, thank you to everyone who's joined us online. You are all most welcome. This evening commemorates the life and work of our founding Dean, Professor Douglas Charles Blood, Doug, as he was affectionately known, a truly visionary academic leader who shaped not just Melbourne Veterinary School, but the modern veterinary profession. Doug's is a unique legacy, and he continues to have a profound effect on the practice of veterinary medicine to this day. In fact, more than 60 years on, his seminal 1960 textbook on large animal medicine, now in its 11th edition, and just think how few textbooks are still in print and in current copy 60 years on. His book continues to be the standard large animal medicine textbook used by vet students and practitioners all around the world. You will not go into a vet school library and not find Blood and Henderson in there. I still have my student copy of Blood and Henderson on my shelf in my office. And it was one of the books that defined my undergraduate years. And it's a truly remarkable legacy that Doug continues to educate and shape veterinary students through his scholarship. Doug had a truly remarkable career that spanned the US, Canada, and Australia, and he truly transformed large animal medicine. He was a pioneer in dairy herd health and preventive medicine, and actually produced one of the very first computer programs to assist with bovine diagnosis. He fundamentally changed the teaching of veterinary medicine, and he single-handedly led a shift from the old-fashioned ways of intuition and opinion to an evidence-based approach, which is all structured around a systematic clinical examination, a thorough understanding of pathophysiology, and the rigour of the intellectual diagnostic rule-out. He also introduced concepts of reflective practice and the importance of learning from our mistakes. Now, these were absolutely revolutionary ideas in the 1960s. Today, of course, they're perfectly normal. They're the foundation for the way clinical practice is taught in every vet school around the world. But back in the 60s, he was a true pioneer. But probably his greatest achievement, as Moira has touched on, was the establishment of the School of Veterinary Science at the University of Melbourne in his role as founding dean. The original Melbourne Vet School, of course, had been established as a private veterinary college by William Kendall in 1888. It was incorporated into the University of Melbourne in 1908, and then, somewhat remarkably, was closed by the university in 1928 due to a lack of demand for student places. Hard to imagine that in today's environment. And not surprisingly, post-war, in the 1950s, there was a growing voice from the agricultural industry and the veterinary profession for a veterinary school to be established in Victoria. And in the end, after some negotiating, the state government approved the opening of a vet school by the University of Melbourne. It almost went to Monash, but University of Melbourne got it, and that was approved in 1960. Doug was approved as the founding dean. And he dealt not just with university administration, tight budgets, external stakeholders, planning an entire campus at Werribee, and designing a new veterinary curriculum, but he also set the shape and course of the new vet school for generations to follow. That was an awesome task, and we can only wonder at his achievements. Now, a year ago, we were proud to unveil the busts of Doug Blood and Ken Jubb at the Werribee campus, a project that was made possible through the leadership of Professor Bruce Parry, the Melbourne Veterinary School Society, and the generosity of the 115 donors who supported the project. Doug, dressed in his signature bow tie, now keeps a kindly eye on all of us at Werribee from his position outside the J.A. Gilruth Library. At Melbourne Vet School, we are proud of our history, and we look to the future through our innovations in education, research, and clinical practice. And there is no better example of this than our focus on One Health, the subject of this year's DC Blood Oration. And so it's now my genuine pleasure and privilege to introduce our distinguished guest speaker for the 2023 DC Blood Oration, Professor Jane Sykes. Jane is Professor of Small Animal Medicine at UC Davis and, as many of you know, is a graduate of the University of Melbourne. Her research interests are infectious diseases of dogs and cats, not cats and dogs, dogs and cats with public health significance, and she has an impressive track record of discovery, innovation, and scholarship in this area. The title for her lecture this evening is Of Humans, Dogs and Rodents, One Health and Leptospirosis. Please welcome Professor Jane Sykes.
Well, thank you very much for that introduction. Welcome, everybody. It's just a real privilege to be here. I want to thank Moira for inviting me to give this presentation. It's just an incredible honor. And again, thank you, Josh, for that wonderful introduction. So I get to actually talk about one of my most favorite topics in honor of DC Blood. And I think maybe I got the title wrong of my talk, and I should have rethought it at the time. It really should be including dairy cattle in his honor. And I also noted that, I have a few letters after my name, but it really doesn't compare to Doug Blood's letters. I had to put them in fine print in order to get them onto my slide. So leptospirosis is really a great example of a One Health problem. And I remained fascinated about this disease after leaving the University of Melbourne. And I did my PhD with Glenn Browning, and I owe him a lot. He really helped establish my career and was instrumental in doing that. And he's here tonight, and I want to let him know that. That's really important to me. But it was really my lessons learned here on campus that took me, when I was in Minnesota doing my residency, seeing dogs with leptospirosis that got me very inquisitive about this disease. And I've remained interested in it ever since. I can't seem to stop working on it and doing research on it. So I hope to share that enthusiasm with you tonight. So leptospirosis is a globally important zoonotic disease caused by the spirochete Leptospira interrogans and other pathogenic species. And there's been some recent estimates of the global burden of this disease in people. One study estimated that it accounted for about 1 million human illnesses per year globally, and about 60,000 deaths. There was a study in the US that estimated that it was responsible for about 0.6 hospitalizations per 1 million US people. And those numbers really don't seem very impressive, right? I can stand up here and say, oh, there's thousands of cases, millions of people, but compared with COVID, that's really a kind of drop in the bucket. And so importantly, leptospirosis is a really non-specific febrile illness, and a lot of cases go unrecognized. So it's likely that this is a huge underestimate of the global burden of disease. And of course, 70% of cases do occur in tropical regions. Anyone know what country has the highest incidence of leptospirosis? It's actually Sri Lanka has the highest incidence of leptospirosis, and definitely paddy field workers are really at risk of this disease because they're wading around in paddy fields which are contaminated with infected urine. They've often got abrasions in their feet, and that's how they become infected. And importantly, in dogs, leptospirosis is a significant cause of multisystemic illness. And typically what we recognize when we see dogs with leptospirosis is acute kidney injury, but it is a multisystemic disease. Every single organ in the body is affected. So while we recognize acute kidney injury, really it can involve the brain, the lungs, the gastrointestinal tract, the liver, the pancreas, and so on. And we still continue to see outbreaks of leptospirosis in dogs, despite the fact that we have effective vaccines in some parts of the world. It really is a disease that could be preventable, but when thinking about prevention of leptospirosis and understanding the epidemiology of it, you really need to be thinking about it as a One Health problem. You've got to be thinking about the environment, as well as the host species, as well as the pathogen. And importantly for lepto, when we talk about the host, we've got to be thinking about the hosts because these organisms are being transmitted among multiple different species of domestic and wildlife animals. So the taxonomy of leptospirosis has undergone considerable revision in recent years, and I'm going to just touch upon it because it's important for understanding epidemiology. So we now have 68 different genus species of Leptospira, and the number grows every month. There's more and more organisms every month that are described, and they've been recently regrouped into two clades, the pathogens and then the saprophytes. And within the pathogens, we have the two subclades, the P1 and P2 pathogens, and then we also have S1 and S2 saprophytes, which are in the environment and do not cause mammalian disease for the most part. And so the most important pathogens are Leptospira interrogans and Leptospira kirchneri. They're responsible for most human disease and definitely most dog disease that we know of. And within those two pathogenic species and other pathogenic species, we have hundreds and hundreds of different serovars. So you may be familiar with these Leptospira serovars. They're classified based on their outer lipopolysaccharide antigens. And then the serovars are grouped together based on relatedness of those outer lipopolysaccharide antigens into serogroups. But this taxonomy, the serotyping classification method is really challenging because it's confusing. There's been a lot of misnaming of leptospiras over the years because of reagent issues. And importantly, because it's just based on outer lipopolysaccharide antigens, you can find the same serovar in multiple different species of Leptospira, right? So they can be genetically very different organisms, but have the same outer lipopolysaccharide antigens. So there's not just one Grippotyphosa, there's multiple Grippotyphosas. You can find Grippotyphosa in interrogans, as well as also in kirchneri. And that's led to a lot of confusion. In addition, for every serogroup, there's there is a serovar with the same name. So we have a serogroup Gripotyphosa and a serovar Gripotyphosa, and that also leads to confusion. So this has led the Leptospira world to move more towards sequence typing and genetic typing methods for studying epidemiology. But the problem is in animals is that we're still dealing with vaccines that are serogroup and serovar specific. So we still need to know what serovars are out there, because in order to design effective bacterins, we need to kind of have that understanding. And so that actually kind of brings me back to my PhD as well, and some work that I did on with typing of Mycoplasma gallisepticum as a student with Glenn Browning. And I took some of the methodology that I learned as a veterinary student, and as a resident was working with Leptospira isolates from dogs in Minnesota, and doing some of these typing methods that, really cumbersome methods of typing. We're using radioactive material to identify bands, and now we're kind of in a completely different world right now, and we have a core genome, a multi-locus sequence typing method for characterizing leptospires. And the numbers of isolates that are getting characterized using this core genome method is just growing again every month. We've got now thousands of isolates in the database, which is helping to understand the epidemiology of this disease quite a lot. So I really like history of infectious diseases, and I think it's an appropriate time to talk about history of infectious diseases in One Health. And so the severe manifestation of leptospirosis in people is called Weil's disease, and it's named after Adolf Weil. Also appropriate with Glenn Browning having spent quite a lot of time in Heidelberg, and Adolf Weil was there in Heidelberg, and he recognized that there were 4 people that had infective jaundice, and they had very similar clinical signs. And so he was quite sure that these people all had the same cause of their illness, this febrile illness associated with icterus. And he was about to get a promotion, actually, but he developed tuberculosis of the throat, and he lost his voice, and so he was unable to work. And he was also unable to identify the infectious agent. This was very frustrating for him. And then in the early 1900s in Boston, this spirochete was discovered in a pond outside of Boston. And this is Leptospira biflexa, which is one of the saprophytic species of Leptospira. So they described this spiral-shaped organism with hook-shaped ends and proposed the name Spirocheta biflexa for that organism. So that was the first discovery of Leptospira. And then subsequently, Japanese miners or it's really around the same time, these Japanese miners had very similar illness to the people in the late 1800s that Adolf Weil had recognized disease in. And I really love this paper because they describe, in February 1915, we announced the discovery of the spirocheta of Weil's disease. It may be interesting, therefore, to review briefly the reasons for the failure to detect the spirocheta at an earlier period. All investigators search for bacteria. The spirocheta resides in the blood only during the early stages of the disease and is present there in small numbers. And at the period at which fatal cases come to autopsy, the liver is either devoid of spirochetes or they're so few or modified in form as to be difficult of discovery or recognition. And we still have that problem with Leptospira today. But the story goes that when this cause of leptospirosis was finally revealed, that Adolf Weil was terminal with his advanced tuberculosis, and apparently he coughed up blood at this time and died. So it was a very dramatic ending to his life at the time of discovery of the organism. So Adolf Weil was doing what was pattern recognition, right? And leptospirosis is really a pattern recognition disease, often a real combination of characteristic clinical abnormalities that can lead you to think about the disease. And so we've actually been involved in developing a machine learning algorithm now. Crystal Reagan, who was my infectious disease fellow and is now on faculty at UC Davis, gathered a team of data scientists to train machine to identify clinicopathologic abnormalities that should alert a clinician to possibility of leptospirosis. And now the goal is to have this integrated into our hospital information system and pop up and say, did you think about lepto? So if you were to go out and try to get leptospirosis tonight, how would you do it? And my story has changed over the years as I've thought about this more and as we've learned more about the organism. So many people would say, well, go and drink some contaminated water. But what we really know about the source of Leptospira is if you really want a ton of them, you should find a rodent, right? And then don't just drink the rodent's urine, but actually eat the rodent kidney tissue. That's what you should do, because that's where you're going to have the greatest concentration of organisms. And so those organisms are going to be capable of penetrating your intact mucous membranes when you eat the kidney tissue. They're just like a wine bottle corkscrew. So those of you who are drinking wine right now, you'll be thinking about Leptospira spiraling through tissues that way, and of course through abraded skin, again, these paddy field workers that are waiting around in contaminated water sources. And then the organism replicates really rapidly in the bloodstream, and that's the leptospiremic phase of illness. And that's associated with a diffuse vasculitis, and we're learning more about how this organism disrupts endothelial barriers and basically disrupts cadherin and is able to penetrate through endothelial barriers. And that typically leads to, if there's poor adaptation, a lot of inflammation to the organism, you get this acute kidney injury, which is followed by, in some cases, chronic permanent kidney damage. And importantly, we think about icterus and Wheels disease, but it's a cholestatic hepatic injury that occurs with a disease rather than it being a liver failure as such. Usually there's not a lot of inflammation going on in the liver, just as was described in the paper that I just showed you. It's this cholestasis that occurs, and there's disruption of hepatocellular junctions that can be seen on pathology. And then ultimately, if your immune system does a good job of it, it will confine the spirochetes to the renal tubular epithelial cells. And what we're learning now about Leptospira and persistence in reservoir hosts is that it can form biofilm on the renal tubular epithelial cells, and that's what allows it to establish this chronic infection and be shed and be really resistant to immune recognition. And so if you were to eat the kidney tissue tonight, and we're going to keep coming back to this, you could expect to become sick in about a week from today. Typically, the incubation period is about a week in duration. And so really we have struggled to understand the importance of different reservoir hosts in different regions. And we're starting to get a better understanding about this. And really, I think the conclusion has been that rodents are worldwide the most important reservoir host. There was one study that looked at all of the papers on rodent Leptospira infections, and they estimated that about 30% of rodents are infected with pathogenic leptospiras worldwide. That's a lot. That means if you see a rat tonight, there's a 1 in 3 chance that it's going to be infected with a pathogenic organism. And of course, in some parts of the world, it could be higher. It could be 90% in some regions of the world. And Norway rats in particular, brown rats, seem to be really good at being reservoir hosts. And so there's been interest in other reservoir host species as well. And we often think about lepto as being a mammalian disease, but a number of studies now finding pathogenic leptospiras in reptiles and amphibians. So it's not just mammals. Interesting study done in the United States in the Illinois region where they found 75% of these blanding turtle species, which are quite widespread, I understand, in the upper Midwest, were infected with pathogenic leptospiras as detected using PCR. And then some studies looking at cats as well, given that they're predating on rodents, and how likely are they to be infected. And this study from Western Australia got a lot of people interested because they collected black rats and also feral cats, and you would think that they would find more lepto in the black rats, but actually they found a really high prevalence of renal infection using PCR in the feral cats. So got a lot of people interested in doing cat studies. We've actually just finished one at UC Davis looking at cats in Northern California and in Texas. And we found, similar to this Canadian study, about 10% of them were seropositive. We didn't find any evidence of organisms in the urine of those cats. We were not able to get kidney tissue though. But it's not that sort of simple as just saying, well, there's a lot of infection in cats. You've got to know whether the strains that are infecting cats are capable of being transmitted to other host species. And this paper is a really interesting one because it looked at many, many studies that quantified the amount of organisms that were shed per mil of urine in different reservoir host species. And so if you look at rats, on the left-hand side, you can see the log concentrations of organisms per mil. Like it's millions of organisms per mil that rats are shedding. Whereas if you look at dogs, it's more like in the range of hundreds of organisms per mil, and cattle, thousands of organisms per mil. But you can imagine like standing behind a cow and it urinating on you, like you're going to get splashed with urine as opposed to having one rat that's peeing on the ground in front of you. But if you're surrounded by, thousands of rats, and of course your risk of getting leptospirosis is going to increase too. And so, there's been this association made between certain serovars and certain reservoir hosts. So rats have been considered to be the reservoir host for serovar Ictra hemorrhagiae, cattle for serovars like Pomona and Hajo, and dogs, importantly, for serovar Canicola. But what we're learning with these new molecular typing methods, sequence typing and MLST, is that there are tighter associations between certain sequence types and certain reservoir hosts, okay? But we still need to understand these serogroups, as I said, because of the vaccination situation. So this study, I think, really fascinated me. It was a study of people who were traveling to the Seychelles Islands, and there were holidaymakers there, and they got lepto. And so these researchers were curious about where these people were getting leptospirosis from, and they hypothesized that it was rodents that were transmitting infection. So they were able to PCR out sequences from 51 people with leptospirosis. They had 32 PCR positive people. And then they collected 740 rats. And when they did PCR on the kidney tissue from these rats, they found Leptospira interrogans in more than 50% of the Norway rats and in about 4% of the black rats on the islands. But when they did sequence typing, they showed that 2/3 of the people had sequence types that didn't match those of the rodents at all. So there was some other source of infection. So you've really got to dig deeper into understanding what sequence types are there to understand the epidemiology of this disease. So we've had this association between lepto and increased rainfall, and that's been of concern with climate change and severe flooding, concerns especially with the proliferation of homelessness in association with the pandemic, and increased rodent populations, and the possibility of leptospirosis becoming a more important problem. And there's been some nice studies looking at the viability of the organism in the environment, using more novel techniques, more innovative techniques, and showing that it can survive several weeks in freshwater sources. But what seems to be really important is mud. And it also goes back to the ability of the organism to form biofilm. And it can form biofilm on these solid substrates that are waterlogged. So muddy conditions promote persistence of the organism in the environment. And we used to think that it couldn't survive freezing as well, but we know it can just reduce the viability of organisms. They can still survive those conditions and be recultured from frozen tissues. And the seasonality of leptospirosis actually varies depending on where you are in the world. If you're having rainfall all year round, this paper from Purdue showed that in the south central parts of the United States where you have rainfall all year round, you can see leptospirosis in dogs all year round, whereas in other parts of the country, really, you could, like in California, we see most of our cases in January, February, March, after we get some heavy rainfall. And so that brings us to situations where vaccination has not been performed in dogs because it's felt that this disease doesn't exist and the conditions are not right for it in those regions. And so I've been involved in a couple of different outbreaks of leptospirosis in the US that have occurred in regions that are quite arid or dry, and vaccination has not been performed. And the first one was in Pima County in Arizona, where there's actually been an ongoing outbreak of leptospirosis there. And so there's been all sorts of hypotheses as to why leptospirosis is occurring in Arizona. Some people think it's irrigation, but I mean, we've got to be thinking about the fact that this disease can be transmitted not just through contact with environmental sources, but dogs that are actually ingesting rodents or ingesting even lizards could potentially be getting infected that way as well. And so that brings me to this study that we've been doing in Los Angeles County that followed an outbreak of disease that began in the middle of 2021. And this screenshot is from the Los Angeles County Department of Public Health website. They were involved in investigating this outbreak. And there was a large number of dogs that were involved. At least 200 dogs ultimately were affected, confirmed to have leptospirosis. And you can see that most of those dogs had clinical signs of illness. Why is that important? Why is it important that most of the dogs were really sick? Because actually it's hard for sick animals that are mounting a really strong inflammatory response to the organism to transmit infection to other animals. You're more likely to get lepto by contacting subclinically infected animals that have good adaptation, biofilm formation, large numbers of organisms being shed in urine. So this puzzled me, that we had all these dogs that were suddenly getting sick in this part of Los Angeles in Santa Monica. So it started in one particular doggy daycare in Western Los Angeles, a very fancy part of town. You know, lots of very wealthy people putting their dogs in these doggy daycares, all very active on social media too, which influenced this. And so the Los Angeles County Department of Public Health came in and did this investigation. You can see they looked at boarding daycare, and there were two particular doggy daycares in the region that appeared to be the source of a lot of cases, and there was no association with dog park visitation. But there were many animals that didn't go to those doggy daycares, that didn't have any boarding, even though there was a lot that had that. There were many that didn't as well. So I'm going to come back to that. And this is the epidemic curve. Again, they published on their website, and you can see that case numbers at about July of 2021, and then peaked around August 2021, and then started to taper off thereafter when veterinarians in the region started vaccinating, because they hadn't been vaccinating, and the public health department said, you need to start vaccinating dogs, and the doggy daycares were asked to shut down their operations and clean up their environment. And they noted that these dogs that were sick were seroconverting with very high titers to Serovar canicola. And so they made this statement, which was that this Leptospira Serovar canicola is the likely cause of this outbreak. Dogs are the primary reservoir for canicola, not wildlife or rats. As a result, this outbreak is being driven by dog-to-dog transmission of Leptospira. And I was like, wow, I don't know whether that's true or not, but I'm really interested in it. And there were other people saying, well, there's all this homelessness in the area, lots of rats. And we do know that some of the dogs that got sick were seen to eat rats before they came down with illness, like their owners observed them to have ingested a rodent. So as I said, incidental hosts tend to get severe disease, and they tend to shed organisms for very short periods, and it's hard for them to transmit organisms to other hosts. So that didn't seem to fit with this picture as well. And so you're, as I said, more likely to get leptospirosis if you're working with healthy animals If you're doing work with livestock, with wildlife trapping, for example, working in water-intensive crops like banana plantations, rice farming, fishing, and working in sewers, as if that's not bad enough, you can get leptospirosis doing that. Military operations involving water. And of course, there's been a number of large outbreaks in association with open water competitions. If anybody likes open water swimming, maybe less so after this talk, but kayaking and canoeing and fishing, as I said. And then there's been a lot of concern with recent popularity of ecotourism, adventure racing, outbreaks of lepto occurring in association with that. So this paper from Japan was an interesting one as well, and sort of similar conclusions made. They studied dogs with lepto, and they had a control group, and they showed that getting leptospirosis was associated with walking along this riverbed. It increased the dog's risk by 13-fold. And they noted that many dogs seroconverted with high titers to Serovar australis, and so they thought that maybe this was caused by Serovar australis and that their vaccines needed to include Serovar australis. So that brings me to diagnosis. So you've all gone out and eaten kidney tissue tonight, and in a week from today you get sick, and so you go to your doctor and you say, well, I think I have lepto because I did this. And they're going to go, okay, well, we'll test you for leptospirosis. And so they're going to collect your blood and they'll send it for PCR testing typically. And then they'll also send serum to a diagnostic laboratory, and in the US it's to the CDC. And they'll do serial dilutions of your serum, and then they'll react to each of those serial dilutions of serum with a battery of live leptospiral serovars. So this is the microscopic agglutination test, which is the gold standard test for diagnosing leptospirosis. In people, at least in the CDC, their panels include more than 20 different serovars. So you can imagine how challenging doing that test is. It is a really challenging, cumbersome diagnostic test to do, and they're working with live Leptospira serovars because they're looking for the serum dilution that causes 50% of organisms to agglutinate as determined using dark field microscopy, which you can see on the slide here, these clumps of organisms. You can see 50% of them clumped together, and they need to see the movement of the organisms in order to do the test properly. And so there's a laboratory hazard risk as well working with these pathogenic organisms, and the need to maintain all of those organisms is expensive as well. So, but it is the gold standard, and by including all of those organisms in the panel, the goal is to have as many as possible so that you can increase the chance of detecting an antibody response to any one serovar, and you may see multiple positive titers, and that's due to cross reactions among those serovars. So unfortunately, in veterinary diagnostic laboratories, we usually are only testing 6 or 7 different serovars, not 23. And so this is a 2-year-old Kelpie. I had a Kelpie in there just for Australian purposes, that was suspected to have leptospirosis. And this is actually Jan Ilkew's dog, if anybody knows Jan Ilkew. She had a Kelpie in Davis. And so you can see pretty much negative titers on day 2 of illness with a low positive titer to Bratislava. And then what we need, right, is a convalescent titer to see a change in titer over time, because this is an acute illness, and you, by day 5 to 7, have not had a chance to make antibodies yet. So on day 10, we recheck titers, and now we have a titer of 1 to 6,400 to Bratislava and lower titers to others that again represent cross-reactivity. And we're really tempted to say that this dog is infected by Serovar Bratislava, right? We just want to do it because we want to make a diagnosis. And so what if I told you that now the laboratory added Pomona to the test? And now you could go on and on, right? 23 different serovars in human medicine, sky's the limit. You could see titers of 102,000 or greater. And so this is not a diagnostic test for a serovar. It's a diagnostic test for leptospirosis, but it doesn't tell you the identity of the infecting serovar. And furthermore, this is one dog with leptospirosis with titers done on day 0, day 28, and then day 78 of illness. And you can see the organism with the highest titer actually changes over time. And when they do those 23 serovar panels in people, they've showed that it's the more convalescent one that's actually more predictive of the infecting serovar than the earlier ones. So the only way to really know what serovars are there is to culture and serotype. And the Japanese have done a really good job of this. This study, they looked at 83 dogs with lepto, and lepto is really hard to culture. You need special media, long incubation times. So they were able to culture leptospires from 45 out of 83 dogs. And they showed that hebdomatis was the most prevalent serovar, followed by australis. And so that's helpful information for designing vaccines. So we've got a lot of disadvantages of microscopic agglutination testing. It's often negative early on in the course of illness. And we have a lot of widespread subclinical exposure. So you do a test and it's positive. Well, it may be that the dog has ibuprofen toxicity, but just happens to have an antibody titer to Leptospira. So positive results are not predictive of causation. So I say that if you're going to do testing, you kind of need to commit the owner to doing acute and convalescent phase testing. Even a single titer is not useful, even if it's pretty high because of previous recent exposure and in some cases vaccination. So we now have point of care tests for lepto, and this is making the issue even more difficult actually, because these tests are just antibody tests, and because they're done at point of care, there's often a lot of pressure in a clinical situation. You've got a dog with acute kidney injury, you do one of these point of care tests and it's negative, and you say it's not lepto, so we don't need to worry about lepto. But it's just that the dog hasn't made antibodies yet. And so same principles apply here. And then if it's positive, it's like, oh my gosh, it's lepto. But it could be just that that animal happens to have an antibody titer from previous exposure or vaccination. Thank you. The one test that's out there that does seem to be proving useful is the Zoetis witness test that's available in the US. I don't know if that's available here, but it's an IgM-based test. And when positive, so far, what we're seeing from studies coming from different parts of the world is that when it is positive in conjunction with compatible clinical signs, usually those dogs truly do have leptospirosis. So we also have PCR tests, and we're really trying to emphasize early use of PCR when antibiotics haven't been given yet, because once you start using antibiotics, they typically go PCR negative pretty quickly. And the problem with doing PCR in urine, of course, is that we know that healthy dogs can shed pathogenic leptospires. And so just because you find pathogenic leptospires in the urine, it doesn't necessarily mean that they're the cause of disease. There was one study done in shelters in the US where they showed that over 10% of around 200 healthy dogs had pathogenic Leptospira DNA in their urine. So that brings us back to the Los Angeles situation, which is like, I felt like we really had to culture this organism to know what serovar we were dealing with, which of course doesn't necessarily prove that it's being transmitted dog to dog. But the first step was that let's make sure that this really is serovar canicola, because everything's been based on antibody testing, and we know that's not predictive of the infecting serovar. So we teamed up with Jarlath Nally, who is a bovine Leptospira researcher at the USDA in Ames in Iowa. And his technician has developed media that are really efficient at isolating lepto. So within 24 hours, they're getting growth of lepto, more like what you would see with, E. coli or strep. And so we had practitioners in West Los Angeles inoculate transport media at point of care. The samples were sent to the USDA, and they were sending us videos like this one within 24 hours of receiving the samples. So the first dog that we tested was positive. It was fantastic, and I can watch this video so many times. It's so fun. You can see the little organisms like zipping around in there. Look at that, and the one that's like heading off out on the left-hand side there. It's really fun. And so we then were able to serotype this organism and show that it truly was canicola. It really was canicola based on serotyping. And we've also collaborated with researchers at Northern Arizona University in Flagstaff, and they've done whole genome sequencing now on multiple organisms from this outbreak. So we've got whole genome sequences, and again, they're aligning with whole genome sequences from other canicola serovars too. So that's been really interesting. So the next step is this really getting transmitted dog to dog, or are there rodent sources of infection? And we know that that now there's studies that have shown that rodents can shed serovar canicola and can be reservoir hosts for canicola as well. So it's just not as simple as saying it's all about dogs being reservoir hosts for canicola, and horses too. But the role of the reservoir versus the incidental host, actually it's been very interesting looking at the California sea lions, because they get leptospirosis and they have outbreaks of leptospirosis, and it's the juvenile sea lions that get really sick. But the adult sea lions act more like reservoir hosts. They chronically shed the organism. They're not affected clinically. And then they have die-offs of these juvenile sea lions seasonally. And so it may be the fact that maybe their immune system is less mature, or maybe there's stress, overcrowding, and that's contributing to these outbreaks. So it's important that veterinarians have a high degree of clinical suspicion for this disease. And I mentioned pattern recognition, and there's a number of different clinical signs that we have to think about with leptospirosis, and these patterns of abnormalities can be very helpful. This is a dog from the Los Angeles outbreak here that had necrosis of its distal tongue in association with vasculitis, and you can see how severely its tongue was affected. Apparently the dog wasn't too painful, remarkably, but they were inspecting the dog's tongue and the whole distal portion of it just fell off. So it was totally necrotic. And we've seen a number of dogs with severe pancreatitis due to lepto as well, so they present predominantly with pancreatitis. And we've recognized a number of dogs with pulmonary hemorrhage syndrome, which has been reported in humans as well. And now it's pretty much standard practice for us to take thoracic radiographs in any dog suspected to have leptospirosis. And because it can be subclinical, and you take radiographs and you can see radiographic abnormalities even if there's no respiratory distress. But you can see the variations in the severity of pulmonary hemorrhage syndrome here. These are multiple different animals that all had pulmonary hemorrhage syndrome. So really, I just wanted to also let you know that we have really just finished a consensus statement update for ACVIM Leptospira consensus statement update, and we presented it at the recent ACVIM forum. And one of the things that we did was we created the first case definition for leptospirosis in dogs, which is a case definition for lepto in people. And this is one of the first case definitions for an infectious disease in companion animals, period, that we're aware of. So it combines diagnostic test findings with clinical abnormalities, and we're classifying dogs as probable or definite cases of lepto. And then on the subject of vaccination, we've gotten several studies now that have shown that vaccines are reducing disease in dogs, but the problem is that we need broad serovar vaccines. And once 4 serovar vaccines started to become available in the US, we stopped seeing leptospirosis in dogs. And so now all the dogs that we see with lepto are unvaccinated dogs. When we had just 2 serovar vaccines, we continued to see disease in vaccinated dogs, but we're not seeing it in dogs that are vaccinated with 4 serovar vaccines. And we know that the new vaccines that have been really cleaned up have now adverse reaction rates that are very similar to distemper hepatitis parvo vaccines. So we felt very confident, and we were in consensus that all dogs should get vaccinated for lepto because it's a zoonotic disease. It can be severe or fatal despite treatment, and exposure can occur regardless of age, breed, geography, or lifestyle. And so despite the fact that this is a non-core vaccine by some groups, we're now kind of recommending that all dogs come in annually and be vaccinated for a lepto. We have cases like this little cute German Shepherd that we're writing up right now as a case report, but she developed severe leptospirosis at just 3 months of age, and she needed dialysis, and her bill was $20,000, which is pretty standard for dogs that get dialysis at UC Davis. So that's kind of the earliest you can vaccinate dogs for lepto. And so in terms of treatment, there's been some debate, especially with people who are more advanced in their illness. So if you delayed going to the doctor in a week from now and you kind of waited around and got a bit sicker, there may be no benefit to going on antibiotics in terms of your outcomes. So you should go as soon as you feel sick. Just giving you that advice now. And the best antibiotic, honestly, for removing organisms from the kidney, based on hamster models, is doxycycline. The problem is that many animals are sick and can't tolerate doxycycline initially, so we treat with penicillins, and then once they are not vomiting, we'll move over to doxycycline. And we do still have precautions for our lepto-suspect dogs, which is a lot because these are dogs with acute kidney injury typically, and we see a lot of dogs with acute kidney injury, but only a small proportion of them are dogs that truly have lepto. And so now with some studies that have really shown low risk of human illness following contact with dogs with lepto, we've recommended in the consensus statement that precautions only be used for the first 48 hours of treatment, and that makes things a lot easier in terms of handling these animals in hospital. And so one of those studies was done by the CDC when they investigated the outbreak in Arizona that I told you about earlier. And they looked at animal caregivers and dog owners that had contact with dogs that were known to have lepto in this outbreak. And many of the animal caregivers had considerable exposure to dog urine. They weren't wearing any protective gear. Many said they got splashed in the face with urine. And so no precautions in many situations. And several people got sick afterwards and thought they had lepto, but they did serologic testing on all these people and they were all seronegative. So they concluded, that was with one of those 23 different serovar panels, they concluded that there was a low risk of getting leptospirosis based on this study from high-risk exposure. And so because this is a really One Health talk, I wanted to show you this really interesting study that was a collaboration between Emory University and the University of Minnesota, where they modeled leptospirosis in a resource-poor area, considering rodents and dogs and people and two pathogenic strains of Leptospira. So it was a modeling study, it wasn't an actual field study. But they identified dogs in two groups as owned dogs or as stray dogs. And so if you're interested in epidemiology and modeling, the study is just kind of fascinating because they showed that transmission of the pathogen to people was mostly likely due to rodent exposure and not due to dog exposure at all. But if you vaccinated the dogs, because there were organisms moving between dogs and rodents, you could actually reduce human infection based on their model. So I think I'm out of time. I could probably talk for 3 hours on lepto. It's such a fun and interesting disease, but I'd love to take any questions, and thank you again for this incredible opportunity.
Jane, thank you very much. That was a beautifully constructed and beautifully delivered talk. We have time for questions. For those of us in the room, if you raise your hand, a roving mic will appear. So please wait for the microphone to get to you. And for people online, please put questions in the chat box and Amber will filter those in. And the plan is to alternate between questions in the room and questions online. So whilst people are thinking of their opening questions, Jane, I'd love to dig in a bit more about your opening comments, in fact, because I, for one, have always found the classification of Leptospiras pretty confusing, and it was really good to hear you work through the complexities of the current classification systems, and also incredibly interesting to see how that complexity translates into practical difficulties when it comes to diagnostics and interactions and understanding what the different diagnostic tests mean. So I wondered if you could reinvent all of this, would you have a single best way of typing or classifying Leptospiras, or are we going to be stuck with a complex mix of sequence typing and serotyping and so on?
Well, if I could reinvent all of this, and I actually think we're nearly there, it would be to have a non-serovar specific vaccine. And there are people who are working on those right now, and they're getting pretty close to availability. So we're going to be having broad vaccines that are not serovar-related immunity, and then we can just move completely over to molecular typing and not have to worry about serovars at all. That's how I would reinvent it.
Yeah, and with molecular typing being so fast now, that would be a very practical proposition.
Yes, and the group at Northern Arizona University have developed, as I showed you in that paper that they recently published, a method for enriching organisms without having to culture them and then doing whole genome sequencing. It's still $1,000 per isolate right now, so it's still really expensive, but it's going to get cheaper for sure. So that will be we'll learn so much about this disease once that happens.
Excellent, thank you. Yeah, we have a question over here.
Jane, thank you. That was terrific. Is there any evidence that serovars of lepto compete with each other? And if they do or don't, how likely do you think that mixed infections are?
That is a really great question. Do I need to repeat the question for the audience, or are we good? Okay. So I don't know about competing with one another, but I do know that mixed infections can occur. And so just me saying that those multiple positive serologic reactions are cross-reactivity, that's a little bit of an oversimplification, because there have been cases described in people where people have been infected with multiple different serovars simultaneously.
So let's go to an online question, please, Amber.
So Charles Galt, she's got a great question. And first of all, I'd like to thank you, Jane, for a fascinating presentation. The media here in Australia is reporting severe shortages of canine vaccine for leptospirosis and fears of the possibility of the disease outbreaks. So would you like to comment on the situation and how the disease may be managed, especially in rural situations?
Well, that's an interesting question, and not working here right now, it's my understanding that the vaccines for dogs in Australia are 2 serovar vaccines still. Is that correct? Yeah. So the problem is that even when we've got 2 serovar vaccines, I think disease can still keep occurring anyway. So that's one aspect of it, although it depends what serovars are circulating here and whether there's been any reduction in case numbers in association with vaccination. So that's something I can't answer because I don't know the answer to that question. And in terms of what to do to prevent it, I think that unfortunately dogs are dogs and they're still going to get exposed a lot of the time no matter what you do. You can control rodent populations as much as possible, but it is difficult to stop a dog being a dog.
Thank you. Let's go to Glenn.
Thanks, Jane. That was fantastic. In your talk, you sort of suggested in the LA outbreak you were expecting an incidental species or an incidental strain, but it turned out to be Canicola. So why do you think Canicola was the cause of that outbreak, and why was the disease so severe?
So my thought on that, and especially there's been now, I think, 3 publications that have shown. There's been one that's shown populations of rodents acting as reservoir hosts for serovar canicola. There's been a study in horses showing that they can be reservoir hosts for serovar canicola, and there's been a study in cattle now, Sri Lankan study showing that cattle can be reservoir hosts for serovar canicola. So I think we've oversimplified it, honestly, and I think that there are other virulence factors that create the incidental host, reservoir host phenomenon, that it's not lipopolysaccharide based.
We've got more questions in the room, but let's take another online question first.
Great. So Vito Catella would, again, great talk, Jane, was wanting to, speaking of One Health, we know that most pathogens follow similar transmission pathways. Do you think it's feasible for future intervention or prevention strategies to take these commonalities into account and be developed in a way that allows us to monitor and prevent multiple pathogens within a more comprehensive One Health framework?
Well, that's a very thoughtful question, actually. So I think that I'm understanding that the question is can we be thinking about transmission mechanisms and patterns, and thinking about those and applying them to other infectious diseases as well, and trying to interrupt transmission by understanding One Health. And I think the first thing is kind of a basic thing, which is to actually think about One Health in the first place, because there's still so many examples of where people have tunnel vision and they're not thinking about multiple animal species being involved, the environment being important. I mean, COVID taught us that. And so I hope that we learned a lesson, and I'm a little bit worried that we still haven't. So I guess that would be my answer, is I feel that a One Health approach is needed still for many different infectious diseases.
Right, we have a hand up over there, I think. And I think there's one over this side, Eddie. Yes.
Thanks, Jane. Wonderful speech. Just had a question about vaccine resistance. With the reservoirs existing in other animals whose owners may not have vaccine hesitancy, or in rodents, do you know the rates at which the bacterium becomes resistant to the vaccines, and how often they have to be sort of renewed, similar to the flu vaccine, that has to be redone and the dogs vaccinated?
Yeah, so that's a good question too. There's, if you look back at the history of leptospirosis in dogs, it was thought that before leptospirosis vaccines became available, icterohemorrhagiae and canicola were the main causes of canine illness, which is also interesting that canicola was one of them. And so that's why we started off with bacterins containing those two serovars. And then we started to see disease in dogs that were vaccinated with two serovar vaccines. And it was thought that that was because we had maybe selected immune pressure, and that there were new variants that were causing disease. But it again is more complicated than that, because if you go through all the literature, looking at all the studies, many of the older studies only included canicola and icterohemorrhagiae in serologic testing. So they didn't have a panel that was broader than that. So recognition of some of these other serovars was in association with inclusion of more serovars in the MAT test. So it may have been that they were around all the time, they were only just getting recognised. So I don't think that we know the answer to that, unfortunately. And then there's also been suggestions that with people spreading into more rural areas, for example, reforestation and more contact with different animal host species, that dogs are getting exposed to new serovars that they wouldn't have otherwise be exposed to, and therefore the vaccines are not working for that reason too. So we don't know the answer to those questions.
Thank you. We've got one more online question, then we'll take this question in the room after that.
So Nadika would like to know, thank you again for a great presentation. Would you be able to comment on the AMR incidences in Leptospira species, especially those from areas in South Asia? Do you see any differences to species isolated in the US?
So I don't know about antimicrobial resistance in Leptospira from Southeast Asia, but I know that we have not identified as such any resistance in, that there haven't been any really very many isolates made in the US, but in terms of what we know about doxycycline resistance in Leptospira generally, there's not been evidence. There have been studies looking at that more globally, and it hasn't been shown to be resistant.
Okay, well, let's probably got time for two more questions, I think. So let's take your question.
Hi, hello.
Hello.
I don't know if this is a weird question, but you mentioned how Sri Lanka was one of the biggest countries with Leptospira cases, but I wanted to ask, because in 2004, the Asian tsunami impacted countries like Sri Lanka and Indonesia, and aside from the damage it caused, it also caused one of the biggest outbreaks of Leptospira in history and in the world, affecting humans and animals. And so given the fact that there are like a multitude of serovars, not only in one species, but, you know, add humans and other animals impacted with that, with obviously in 2004 we didn't have the technology that we have now. But if, God forbid, it was to happen again or in the future in an ideal world, how would we manage something like that?
Yeah, and I think vaccines for people in regions where leptospirosis is prevalent, and there is some vaccination of humans that goes on in outbreaks and in hyperendemic regions, so it's not that there's no vaccination of people, but we're getting closer to having really good vaccines, broad protective vaccines for Leptospira. Joe Vanetz in the US is doing some really interesting work with that right now. So I think it's going to happen. So I think vaccination would be the way forward.
Right, let's take your question, probably the last question for this session.
Fantastic. Thank you very much. Jane, thank you very much. Making leptospirosis cool again, so thank you.
It was always cool.
Always cool. I work in general practice, and so I've diagnosed one of the cases of leptospirosis in Melbourne that's been... so I often get an email from the DPI saying, how many leptospirosis cases? So I'm trying to work out, do we vaccinate? Do we not vaccinate? How much do the puppies hate having an extra vaccine, and all these oodles that hate getting needles? So trying to give them an extra 2 when they're young, to try and protect is always a pain in the bum. My question for you, though, is given the one world aspect and the human health, the zoonotic things, it's always a pain when you've got a dog that you think could have lepto and you've got to try and say to the owner, I'm really worried your dog could have lepto. We need to do tests, but it's going to cost you and it's going to be expensive. Mm-hmm. So you're kind of guilting the owners into saying, we need to run this test because we've got to see if you and your family are going to be okay. Could you comment on how good it would be if the Department of Primary Industries, the government, would actually say, through your algorithm where you had, of going, hang on, you've flagged a couple of things here. You qualify for a free test to be able to see whether or not your dog's got leptospirosis. What test would you choose? And do you think it's a good idea? And do you know anyone who can get it going in Victoria? Because that'd be awesome.
Well, so that's I could probably give a talk on for an hour because, first of all, what is the real risk to an owner of a dog that might have the disease? We still don't really know, but we know it's lower than what we previously thought. In the consensus statement, we actually discussed the possibility of dogs that present suddenly with acute febrile illness, where leptospirosis should be on the differential diagnosis list, given that if you wait 1 day and don't treat that dog, and it comes back to you the next day and now it's in AKI, and the outcome for that dog is instantly going to be poorer, it's going to be more expensive, there's more zoonotic risk. We talked about one of the recommendations, if you see a febrile dog, this is really controversial from an antimicrobial stewardship standpoint, treat that dog with doxycycline. I would say that's less controversial than all the doxycycline therapy that goes on in the United States for dogs that have positive antibody tests for Borrelia burgdorferi, which is just bucket loads of doxycycline in some practices. But we talked about that. And then the testing situation, if there was just one test and it was an acutely sick animal, it would probably be a PCR test, not a serologic test. But you really need to combine both types of tests, ideally. I don't think I answered your getting it all paid for question.
Well, Jane, thank you very much. I know you could have talked for hours on the subject. The fact that our audience, I think, would ask questions for hours is a measure of how engaging your talk has been. So thank you very much indeed. Please join me in thanking our DC Blood Oration lecturer.